Okay, welcome back for the second part of the genome variant analysis section. Today we're going to be looking at how to do read mapping and variant calling. Okay, so in the first part we cleaned up our reads. So if you haven't finished the first part, then you need to go back and do that first, because what we're going to map are our trimmed reads that were the output from part one of this variant analysis section. And so what I'm going to do is go through a little bit of background on read mappers, and then most of today will be tutorial. So we're in this section on sequence analysis in terms of an overall, what you might do in a genome sequencing project. We did quality control in part one. Today we're going to map the reads and identify the variants. We're going to do both of these. Okay. So first question, what are we going to map the reads to? There either is a reference genome available, or you're going to have to do de novo assembly of a genome. Today, what we're going to do is a reference genome, but if you're working in a non-model system, or you're developing a new organism, you may have to do a de novo genome assembly first. So we're not going to talk a lot about that. You may get that in the genomics course next semester. So where do you get your reference genome? NCBI, the obvious place, or that's your reference, your main site. That you could also potentially get that from Ensemble. We talked about those databases before. If you look in their genome database, that's where you're going to go. So actually, I realized I forgot to share my screen. Okay, so, right, jump back a few slides. We're in part one. This is what I was talking about. We've done quality control. Now we're going to map reads and identify variants. And what do we map the reads to? Either reference genome or de novo assembly. Go get your reference genome at NCBI. Could be you go to Ensemble, one of the European or Japanese databases, but in principle, you should, if it's available, you should be able to get it from the NCBI genomes database. The option two, de novo assembly. You can sequence the genome. This is still not anything, especially if you need to get a high quality assembly. It's getting better. It's still not cheap, but some of the technologies I talked about in the last class period, particularly the long read sequencing, Hi-C, which is the Illumina method, where you get contact maps based on physical distance, that makes the assembly better. Also using a combination of PacBio, which is the long read sequencing that I talked about first, or Oxford Nanopore, but typically it's PacBio because they give longer reads. And the problem with PacBio, remember, is that it has a high error rate. So if you couple that long read but high error rate with Illumina data, which gives you tons and tons of reads and low error rate, you can sort of use the long read PacBio as a scaffolding and then line up, do read mapping to line up the Illumina data to fix all the errors. So some strategies, making de novo considerably better than it was a few years ago, but it's still not cheap and it is laborious. The analysis, the assembly part can be tricky. The other thing that you might be doing if you're just interested in transcriptomes is you may just do an RNA-seq project and then do de novo assembly of those transcriptomes, and then do that transcriptome becomes your reference. So you don't have the whole genome, you just have the expressed genes, but then you can go back and align reads to that. And in an RNA-seq experiment, it's the how many reads align gives you some indicator of how much was that gene being expressed. And then finally, you can add on to, you've just generated some new sequencing data. You can add on to a low quality public reference. So your NCBI genome for your organism isn't very good. You can try to reassemble by adding your data, especially if their genome was based on Illumina and you have mate pair or PacBio data, right? You can take that public one and try to just improve the assembly. There's lots of reasons that can be messy. The main one is if you don't have the exact strain or the exact inbred line, whatever the case may be, same population, differences between what you sequenced and whatever the current or publicly available reference are going to potentially cause additional problems in the assembly. Okay, so that can be a messy process. So what do I mean in part by low quality public genomes? Genomes come in all different qualities, right? So the main problem with resequencing and mapping is if the reference genome is highly fragmented, which means that it's not in, I think most people intuitively imagine the genome sequence of something looking like chromosome length DNA, and that's just not the case. Usually it's broken up into the pieces that you could assemble. And especially in model organisms, you at least know how those pieces that you're confident in assembling are related to each other linearly. And so you can place those pieces in order, but you still have gaps. Those gaps are caused by lots of reasons. You have highly repetitive sequences that you can't, especially with short read sequencing, you just can't figure out exactly what that string of DNA should be. And in many cases, how long that is from other methods besides sequencing, right? Recombination rate or something like that. But you still don't have the physical sequence that links those two contigs or scaffolds. Now, I've thrown around the words contig. Contig is the sort of shortest assembly of reads that you're confident in that sequence. Then you scaffold contigs together. Usually, if it's called a scaffold, there's less confidence in right. You might have some contigs in there that you don't know the orientation, but they're built into a larger scaffold. And then usually, when scaffolds get to be chromosome size, you're talking about a chromosome. Okay, so scaffolds can be up to chromosome length. Some of the problems with so mapping and fragmentation are a problem. The other problem I mentioned when you're a minute ago talking about how trying to add on to a public genome with your sequence is problematic. Well, it's the same problem if you're trying to map your data to a strain or species or population that is not the same one you sequenced. The more distantly related, the fewer mappable reads you're going to have, right? Because the sequences don't match well enough for your read to find a place in the genome. And that also means, right, if you accidentally map a read to the wrong place in the genome, you're going to call variants that aren't real. This is analogous to the orthology problem, right? The reciprocal best blast hit, except now everything is one way. You have your read and you're trying to figure out where it is. The more evolutionary distance between the thing that you sequenced or your reads and the thing you're trying to align it to, the more potential you have for errors. And right, the fewer reads that may map. Finally, this note on annotations. If you don't have annotations, right, and by annotations I mean functional annotations, right? Where are the genes? Because you sequence and assemble a genome, you're still a long way from knowing where the genes are in there. Without an annotated genome, it's hard to know what the mutations mean for the biology of the organism. With annotations, you have some potential insight into the effects of a mutation, particularly if you, again, you can tell what is coding and what is non-coding. And in the best cases, right, the reason model organisms are model organisms is that there's lots of functional data. So even in non-coding regions, you might know mutations in transcription factor binding sites and things like that, that help you make predictions about if you call a variant, what is the effect of that variant, or what might be the effect of that variant. I don't know why that's, there we go. So BLAST, talking now about how do we actually map the reads. I talked about this a little bit in part one, but the BLAST is default BLAST is too slow for next gen sequencing scale read mapping, where you've got millions, potentially billions of reads. There are lots of aligners that have, or mapping software that have grown up over the years. The one we're going to use is here, Bowtie 2. That choice is partly based on ease of use and the fact that it's installed on bridges for us, so it's easy to deploy. Okay. In general, your choice of aligner is partly based on the sequencing technology. We have Illumina sequence. Bowtie 2 is designed to handle Illumina sequence. So, right, it knows the quality scoring and all that kind of stuff. It knows the bias that are there. It knows how to deal with paired end sequencing, right? Also matters whether you're sequencing DNA or RNA, right? Because if you're sequencing RNA for an RNA project, the introns are going to be spliced out of most of the RNA sequence. So when you go to try to align a read where the actual sequence spans an intron, that's not going to map to the genome if you use an aligner that assumes you're trying to map DNA to DNA, right? So choosing your aligner depends on the kind of experiment you're doing. Okay. And then there are some fundamental issues that none of them get around. There's always a trade-off between accuracy and speed. And then how much time do you have on whatever the hardware that you're running it on? Okay, we don't really have to deal with these so much. I mean, they're a consideration if you're running it locally. But like I said, part of the reason we're going to use Bowtie is that it's actually on bridges too. And it's sort of been optimized for that environment, so we don't have to worry so much about the limits on CPU time, at least for the data set that we have. Now, one consideration is if you have a really big resequencing project, mapping, what we're going to do today, is relatively fast compared to genome assembly. And so because, like BLAST, it it's looking for exact matches first, right? So let me skip ahead 2 slides. All right, how does read mapping work? It has a similar strategy to BLAST. It's just optimized for short sequences that are expected to match really closely. So it builds an index out of your reference genome. Usually those are, or often they can be pre-computed. We're going to actually walk through that step here in a minute. And in some of the aligners also index the reads. So you can think of what I'm saying, it indexes them. It does something like our pattern to number, number to pattern, that is going to make the searching scale linearly, okay, or as close to linearly as possible. Where, for example, it's only got to read through the genome 1 time for every read. So it'll basically scale with the number of reads that you have, not the number of reads, you know, times the, or raised to the genome size or something like that. Just a few notes here. I'm not going to spend the time to walk through these, but it essentially is working like BLAST, except where it's taking small words, looking for exact matches, and then building out. The main thing to note is that once it's done that, it sort of gives you a it calculates a probability that it found the right spot, and it gives an alignment score. So you get that, which would be equivalent to the BLAST bit score. So you can get a bit score, but in this case, you're also getting the mapping score, the probability that it found the right spot. And those are sort of equivalent to the E value and the bit score, or not equivalent to, they're analogous to. You know, BLAST E value, what's the expected chance that you got this hit? And then the bit score, what is the sort of alignment quality? I skipped over this slide just to highlight the aligners are an active area of research, especially as our ability to generate such huge amounts of data increase. You know, we've got genome projects that are sequencing thousands of genomes, and now we don't want to align individual reads to a reference. We actually want to build whole genome alignments to study genome structure evolution. That is another level of problem, right? If you think back to our multiple alignment problem, those are pretty slow just for single genes. And so, you know, just about a year ago, this new aligner, Progressive Cactus, came out that actually makes multiple genome alignment sort of doable. Well, not sort of, it is doable. So pretty cool tool for looking at genome structure evolution. Partly throwing that in just because this is something that my lab is interested in, and some of the other genomics labs in the department. Okay, so I talked about how mapping works. Mapping scores and alignment scores, that's what I was just talking about. Mapping is sort of analogous to the probability, analogous to the E value. If the probability the read is in the right location in the genome, and then the alignment score tells you how well the whole read maps to that sequence in the genome. We talked about this a little bit in part 1 too, right? When we talk about paired end mapping, there's an expected insert size based on the library prep. So right, you have your reads on the end, and then you have an insert of known length, or at least you know the range of lengths that it should be. And so mapping will use the anchors, use the reads as anchors to a specific location, and then see if the internal spacer is the right distance. And sometimes you can only map one of the reads, and the other one doesn't, and different aligners deal with this differently. Sometimes they'll put it into a different bin, which you can then, depending on how much data you have, and if for example, you might go back into your alignments later, which we'll look at toward the end today, and say, oh, hey, this region of the genome didn't get any reads mapped to it. That's kind of weird when I've got, more or less uniform coverage everywhere else. And then you go into this bin of unmapped reads, okay? And you can take those out and sort of use more specialized tools or pull that chunk of the genome out. These are labor-intensive methods to figure out what happened there, but what is often happening is that there's structural variation. So your reads aren't mapping with the right orientation or the right distance apart because of duplications, insertions, deletions, whatnot. Just some nomenclature here. If only one of the reads in a pair maps, they're known as singletons or orphans. And if they both map, but they're not in the right orientation, a lot of times those are called discordant pairs. Right. All right, so exercise 5, this is continuing on from exercise 4 in the previous one. So we've got some trimmed reads, and we're going to map those to our reference genome with bowtie 2. I think I said that these might be human data in the last section. They're E. coli. I don't know what I was thinking. And that's partly just because this is the data set that the TAC group generated, but again, because it's a small genome, these exercises should go fast enough to finish during class time and in Interact sessions, so we won't have to use Slurm job scripts to do this. Before we get too far, though, I want to make it a note that you need to go get this FASTA file from Canvas. Okay, I for some reason I left it out of the 3 files that we uploaded in part 1. So this is on Canvas. You just need to bring that over to Bridges, and then I'm going to make a new directory to hold everything we do today. Okay, so I'm going to move this over. And there are going to be a few times during the thing today where you might want to just pause the video and make sure you've got everything moved over. So I'm already logged into Bridges 2. Let me go up 1. So when you log in, when you go to your home directory, you have this probably. Okay. The stuff we did for alignment, stuff we did for blast, and then this is the directory that we created last class period, so the GVA data. Okay, and then that tarball is the one that left out the FASTA file for the E. coli genome. So you'll want to go get that, and then when you get it, so let me cd into the GVA folder. Right, so you should have had all 4 of these. Now, I suggest that you make a new directory. I'm going to call mine trimmed data. Oops, that's not it. That's where I actually did my trimming. Oh, it's the bow tie mapping. Duh. Okay, yeah, so we're going to generate a bunch of these files in a second, but the things that you need to move into this directory are the FASTA file. Okay, so go back to the PowerPoint here. You need to move that FASTA file that you just went and got from Kansas into your new directory. I named my directory bowtie mapping, bowtie underscore mapping. And then I need to move my trimmed reads into that new directory as well. So you can see I have my trimmed reads. I pulled those over from the stuff that we did Tuesday. So again, like I said at the beginning today, if you haven't completed part 1, you're going to need to do that before you actually can work on these, right? And this is what I named my trimmed reads. Your trimmed reads might have different file names. That's okay. So once you've got all that stuff in your new directory, don't forget to start your interact session. You don't actually have to start interact to move the files around. Okay, so while we're waiting for that to start up, what are we going to do? We're going to load the module for bowtie 2. In this case, all lowercase. So ML, right, that's the shortcut for module load. Okay, just got an indicator. We've got our job started. Whoops. So ML bowtie 2. Then we need to run this command. So I'm actually going to use copy paste. Maybe. Yes. I'm trying to grab it without getting the enter on the end. Okay, so bowtie build, what this is going to do is generate the index file. So remember I said that most mappers, well, all mappers will index the genome, which is what this is going to do, right? That's the genome file that you just went and got. It has to be in the current directory. And then this is the base name for all the index files that are going to be generated. So it's going to generate a bunch of indexes. Index files. So I don't remember if this generates the FAI file or the BT ones. Yeah, actually this is the BT. Okay. So basically what this is going to do is chop the genome all up and make it machine readable. So after you execute, you should see something like that. And then at this point, you will have generated all these BT2 indexes. Next, we're going to run the mapping. So I've kind of spelled out everything that needs to go in here, except you're going to have to modify it to your name, the names of your files, right? So this -P64 is saying run 64 parallel jobs. That's the most we can run, I think, on an interact session. If we were using Slurm, we could go up to 128 if we got a whole RM node, right? With the RM being the queue on Bridges 2, if you remember that from Slurm lecture. The -X is the index base name. So all of the indexes that we just generated, telling them the name, right? So that has to match that thing. This is a -1, and this is the way to tell bowtie 2 that we're giving it paired end data. So we give read 1 and then a 2 to tell it this is read 2 for the paired. Then we have a -U that says, oh, I'm actually going to also give it the unpaired data. Remember when we did our trimming, we had some where it lost one of the reads because it was not of high enough quality. So we have all of these reads that either don't have a read 1 or don't have a read 2. So we do -u, and then these go in comma separated. So we trimmed reads 1u and trimmed reads 2u. Those are my untrimmed reads. Then -s says this is I want SAM output, SAM file output. We'll talk about what that is in a second. And then the file name, trimmed.sam. Okay, and then here, 2 and the greater sign. This is the write file redirect, where before all we did was just the greater than sign. That outputs the std out, right, standard output, to whatever file you give it, or whatever file you name, right? This file doesn't exist yet. We're going to write it. Bowtie 2 does something really weird where it writes the summary of the mapping to the standard error output, not the standard output. So the std err, standard error, the way to redirect that output to a file is the 2 greater than sign. Okay, so just some notation that we haven't seen before. And then, right, so we're going to send that summary to a file I'm calling summary.txt, but the name is arbitrary. And then I also put an and sign here, the ampersand at the end, because I want this to start in the background. That way I get my command prompt back right away. If I leave the and sign off, Bowtie 2 is going to start running and mapping, and I won't get while it's running, I won't have a command prompt. Now, we could pause the job, send it to the background, all the, way back when we learned Unix job control commands. I think I have that in the next slide. Yeah, so if you don't use the ampersand, this is going to take a while to complete. And so if it just starts running and you forgot to put the ampersand, right, you need to go look at those job control commands. Basically Control Z and then BG will send it to the background, and then you'll have your command prompt back. All right, so this I can copy paste because this has my file names in it. You will have to modify it so that your trimmed read file names are correct for whatever you named them. Okay, and so now it's telling me the job number. Now, how do I see, like, I can't tell what's happening. I don't know if it's working or not. So I'm going to do top-u and then my username. These are Unix commands that we learned way back. This tells me, okay, so my Bowtie 2 aligner is working. It's taking up, just jumped to 102% of the CPU for the node that we're on. So it looks good. That's running. And then I do Q to quit that. If I do LL, I can see that I now have a SAM file that has started, okay, and a summary text that is empty at the moment, but it's been created. So it looks like everything's going okay. When it finishes, it will give me a done line. Okay, so while that's running, let me go through a couple other things. Something to note, right? This is going to take a while. The output SAM file is going to be huge. Okay, it's just a text file, so it is human readable. We'll talk about SAM and BAM because we're going to actually convert some of these files today. Well, we'll talk about when we get there. So the warning here is that these things can be huge, and so you don't want to have this on your local computer and just kind of open it up in a regular text editor, because depending on how big it is, the R1 today is, I forget, it might be, oh, how big are these? 300 meg, so it might be a gigabyte when all is said and done. But this is a small E. coli genome data set. If you're working on even a beetle, right? I have regularly SAM files that are 50 gigabytes. If you try to open that in a text editor, it just crashes your computer or freezes it. So don't do that, right? We've got all kinds of Linux ways to look at that, or Unix ways, right? We can use less or head or grep to look at really huge files. Just check progress over here. If I do LL, you can see it's writing the SAM file as it goes. So it's up to 560 megabytes. Still going. When it's completed, like I said, we'll get a done message, and then something like that is going to go into the summary text. So, but we'll go back and look at that once the job completes. So what is the SAM file that it's writing? It stands for sequence alignment map. It is the file that says for every read that you input, where does it map to the genome? So these are just some useful. It's a standardized file format. So any SAM file you get should essentially be the same. Once you have generated your SAM file, or in a second we'll convert that to a BAM file, you don't need the reads anymore because it has all the information from your reads encoded in the SAM file, which is why it's so big, because it has not only all of your millions of reads, but it has all the information about where they mapped, how well they mapped to the genome. Okay. Are we done yet? Whoop, we're up to 929 megabytes. So in a second, we will look at what that SAM file is. If you're not caught up, you might want to pause at that point. But what you'll see is just sort of like the FAQ files, they have sort of crazy notation because now we've got our header line, right? This is very similar to the header in the FASTQ files, except the @ sign is gone. Then we have our sequence, and this is the quality. So it moved those from separate lines onto the same line, okay? And then, actually, don't remember off the top of my head this notation. This is probably the cigar notation, which I'll tell you about in a second, okay? So essentially, the file format for SAM is, like I said, it's standardized, and it's not often, well, maybe not ever, that you're going to go actually try to read the SAM file, right? The software is going to do that for you, but especially if you're trying to write new software to do some kind of genome analysis, you need to, it's not something you're ever going to commit to memory. You're going to have to go and reference the format in order to know which lines and how to pull those lines out, how to pull that information out. But I mentioned cigar format, so this was one of the cool breakthroughs in developing SAM as a compressed representation of how reads map to a genome. And here I'm giving an example where this read maps to this part of the genome. It says there are 2 base pairs that are soft clipped, right? So the first 2 base pairs on our read don't match. And so in the case where you've got ends that don't match, it will soft clip those, or at least, yeah, that's what does soft clipping mean? It means that they're going to stay in there but the variant caller is going to say those are low probability. So after we do all this mapping, then we're going to do variant calling. So it won't get variants by default. Then it says there are 5 matches. Well, they're not 5 exact matches, right? A, T, G, T, C, right? 3, 4, 5 matches. Okay, so, but those are positions that align well. Then we have 2 base pair indel. So it's calling that an insertion in our read because we've got 2 extra base pairs here. Then we have 4 more matches, G, G, T, G. Then we have 1 deletion in our read. The reference has an extra. Then 5 more matches, and then 6 base pairs that don't align well, and so they're soft clipped. Okay, so all the information about how well, and well, really how this read mapped to this spot in the genome is encoded in a pretty short string. Okay, which, despite being a huge file, it's actually holding a tremendous amount of information. Okay, so I hit enter. Sometimes that done, I actually, sometimes I feel like it pops up automatically. Sometimes as soon as you hit enter back on the terminal, it tells you it's done. So this tells me done and the command name that finished. Do LL. And you can see now we have some information in our summary, and our trimmed.sam is 1.14 gigabytes. So let's use less to look at the summary. And you can see if we go back to this slide, so that's back on the slide. What I meant by once it's done, you can look in the summary text. So that's what I'm doing here. And what is it telling us? It tells us this is how many total reads it tried to map. Okay, so between my paired ends and my unpaired reads, I had 3.5 million reads. This is how many of them were paired. This is concordant alignments. Remember, that's telling us what it means by concordantly is that it had expected distance, like the default, I forget, 200 or 300 base pairs between read 1 and read 2. So it mapped the way you would expect a paired end read to map. And how many didn't basically aligned concordantly 0 times, only 2.5%, aligned concordantly exactly 1 time. These are the ones that these are the good ones, okay? Because they mapped to a unique place in the genome and they mapped in an orientation that is expected. These guys couldn't find a place in the genome to map concordantly. And then these guys, 3%, mapped concordantly more than 1 time. So it couldn't find a unique place in the genome to map those concordantly. Of those ones that did not map, so this category right here, couldn't find a place, it says that 4% of those had discordant alignments. So these are sort of interesting ones in terms of potential structural variation. And then these are the ones that didn't map at all. So it's basically that minus that, I think. I'm doing my math right. This is all just giving you more information about, so there were something like 114,000 of this group that, actually it's not that group. I'd have to go back and look at the documentation. I don't remember off the top of my head what it's doing here with these mates. But then once it gets down here, now it's talking about the unpaired reads. So these are the things we told it didn't have pairs to begin with because of our trimming. And overall, it was only 27.88% of the data, right? So this number plus this number should be 100%. That's all the reads we gave it. Pretty small percentage did not map to the genome, so they couldn't align. Almost 90% were unique, so that's actually really good for single unpaired reads, right? So these are just single end reads essentially now because pairs don't exist. And then 3.27 could not map uniquely. 96.92 overall alignment rate. That is amazing. So that's super good. Right, and a bunch of that are things that couldn't map multiple to a unique place. So that's a really good result when you've got 96.9% mapping, because remember it has potentially, I don't remember in this exact experiment, but they could be different strains, right? So reasons you get all this, the potential structural variation, things like that, could be different strains. I also, now that I think about it, I don't remember if I left in all of those poly A reads. I think I don't remember, but it would be another thing to go look at in the unmapped reads stuff. So that could be explaining part of that as well. So I'll hit Q to end there. And if we go back, where are we? Okay, we just talked about cigar. Oh, we didn't look at the SAM file. So let's look at our trimmed.sam. And it looks a little wonky because I'm going to shrink it down for the moment. So these are individual lines in the SAM file, right? So this is the format we were talking about. So all of that. So that's the top part. I'm going to do tail. Okay, and so you get the bottom part. So it tells you, right, like I said, this is the mapping information sequence, the quality. That's interesting. Okay. I'm going to keep moving here. So the next step is we're going to identify variants. So when we talk about identifying variants, and I've already mentioned looking at structural variation with those reads that map discordantly. Even though it's often referred to as single nucleotide variants or SNVs, in reality, it's not necessarily just single nucleotides, although that's the vast majority of the mutations. And you even get into this gray area about what is a mutation. And if you have a 5 base pair deletion, is that equivalent in some kind of evolutionary counting to 5 single nucleotide mutations? Right? People would say no, right? The indel that deleted 5 or inserted 5 is 1 mutation. So counting the number of mutations isn't so straightforward. But SNVs capture all of that stuff. The real definition of an SNV ought to be, it's a variant small enough that most reads are going to span the variant site, right? So in our case, we're talking about 30-ish to 36 base pair reads. So it can't pick up indels that are bigger than that. In fact, it can't pick up indels that are maybe even half that size, because then the read's not going to map. So the size of variant has always partly been, or the ability to detect the variant has been partly dependent on the length of the reads, so the sequencing technology. So short read sequencing has limitations on what kinds of variants it can detect. Now, like I said, with discordant mapping of paired ends, you can go back and there's software that will try to determine duplications, deletions that are larger based on the paired end information. But it's extra steps, and it has always been a little bit fraught with error. Some of those are going away. Long read sequencing is solving lots of those woes, right? Because if you have 80,000 base pair reads from PacBio, for example, you can get a handle on some pretty big structural variation there. Okay. Yeah. Okay, so next steps. How do we call variants? Well, we need to get SAM tools. So let's blow this up a little bit. I should also say that I've kind of glossed over what all of these are, but I provided a link in the SAM file here. So this will take you to the SAM Tools website, and it has a description of what all the columns are, how to determine what is the XM, XG, stuff like that. Okay. I don't keep that stuff in memory, my own memory, because if I ever need to know, I just can look at the documentation. Most of the time you don't need to know because the software is aware. Right? It's one of those things where once you program it, you don't have to remember it. Okay, so we already have an interact session going. We need to module load SAM tools, and then we need to run this. So here we're going to index again. It's not enough that we indexed it for bow tie. We need to index it for SAM tools. When I pasted that in, I had the enter sign, so it already ran. That's the one that generates the FAI file. Okay, we can look at that one. very small file. It's basically just saying the name. In this case, it says the contig name, but our FASTA is the whole genome. In the FASTA file, it knew this because it was the name of the header. That's where it pulled it from. It says how many base pairs are in it, right? For us, this is pretty big. So all that stuff earlier, this is very confusing, I realize. I call it a contig. In the case of this E. coli genome, they're the same. But there are lots of situations where you could have multiple rows here. Now, because our FASTA file only has one header and it's the whole genome, if you did this to human, for example, you could have one of these for each chromosome. And it's essentially the contig is however continuous length of sequence that is. It's kind of unfortunate terminology going back and forth, but that's just kind of the way biology works. Contig is something where you know the continuous string of sequence. It's not necessarily a length, or not necessarily defined by length. So that's the number of bases, and then some index values. Right. Whoops. We didn't open a FASTA file, but just to give you an idea. All right, so it's saying these are 60 base pairs long, and it just needs to know that so that it knows not to include the new lines there. And bases and bytes are the same in this example file. All right. So a necessary step, but not a whole lot of information. That indexing was did not do the same things as the bow tie did, right? Okay, so next few steps here. I put file name here because now, mine is called trimmed.sam. Okay, and I'm going to keep the names when I convert to BAM, but you'll have to fill in the file name here. I copied those all over to a text editor so that I can copy paste really easy. So you may need to pause here so that you can get these commands worked out. So first one, I'm going to do view, and this just says make the output into BAM format, and then name that output trimmed.bam, and then this is the input file. So do that. Now it's encoding a 1.15 gigabyte file from SAM format to BAM format. BAM format is just turning the human readable SAM file into a binary version that is much faster for the machine to read. Okay. Now we're going to have to sort that thing because if you remember when we did our mapping, the dash P64 spread it out over 64 cores on our machine, all running in parallel. So we were mapping 64 things at a time, okay? And that helped things run much faster. But it also means that as those matches were happening and being added to the SAM file, that they get all out of order in relation to where they are located in the genome or where they were located in the read file. And so what we're going to do is reorder that by sorting the BAM file. And you might be able to see if there's enough here. Yeah, so for example, these are the positions in the genome in our SAM file. You can see they're kind of all over the place. So we're going to convert to a BAM file, and then just because BAM is much more efficient for the machine to read, so that's why we do that step. Saves time and all kinds of this downstream stuff we're going to do. And then there are SAM tools is really a whole package of tools that can calculate stats, and we're going to use it to also do SNV calling. Actually, we're going to use BCF tools. Used to be in SAM tools. The packages to do that SNV calling are still in SAM tools, but yeah, there's kind of it's in a transition period. I guess that's the best way to leave it. If you try to run some of these things in the SAM tools versions, it throws errors to warn you should go over to BCF tools. There's a whole complicated history there that you don't really need to know. You just need to know how it works now. All right, so it looks like our conversion to BAM worked. We can do LL, and now we have trim.bam and trim.sam. Now, this is binary, right? So if I try to look at this, it's going to look like garbage. Yes, I want to see it anyway. See, so it's converted this into machine language. Makes me think I'm in the Matrix or something. Okay, now we need to sort that. All right, so it's got my file names. It's going to sort that BAM file. So one of the things to note also is that the BAM file is way more efficient than the SAM file. Right? It shrunk that 1.1 gig file in machine language. That only equates to 262 megabytes. Note, just so you don't overwrite your original BAM file, that this is the output file I named trimmed.sorted.bam. So that now I can tell my original BAM file from my sorted BAM file. We're going to use the sorted one from now on. And then last thing we're going to do is index that thing. Okay. So SAM tools index that trimmed.sorted.bam. All right, so just check that. And that generated this BAI file. And that last step is necessary so that we can move on to the actual calling of SNVs. Ignore this slide. I don't know why. Okay, I need to get rid of that. That's old. Instead, wow, the numbering is really off. So that needs to be 5, 6. Sorry about this. It'll be fixed in your PowerPoint when you get the PowerPoint for this. Okay, so we're going to do the variant calling with BCF tools. So we need to do module load BCF tools. And then the first step is to generate the genotype likelihoods. So what it's going to go through and do now is take all of our mapping data from the BAM file and say, where does it vary from the reference? And this first step of MPileup is going to just calculate probabilities. So is it correct based on how well this thing aligned? And then the second step we're going to call, and we have to tell it what the ploidy is, and then it's going to generate a file that actually lists all of the variants in the data. Okay, so BCF tools, MPileup. The file that it's asking for -F is with the genome file. Okay, so what's the reference genome? Then this is our sorted BAM file, and then the output in BCF format. So call format is the CF. This is the binary version. Sort of like SAM and BAM. Like BAM is the binary, except instead of having an SCF, we're going to have a VCF or variant call format. We'll have that in a second. Getting ahead of myself. First up, BCF tools MPileup. So we're doing the MPileup. I have a note here, right? It said one sample and one input file. That's right. We just gave it one BAM, excuse me, BAM file. And then it says that the maximum number of reads per input file is set to D250. So I have a note here just to explain. It doesn't mean that it only takes 250 reads out of your file. It means that it only allows 250 to map to any one position. And it's purely a time-saving device. We don't have to worry about in our data set, we don't have sequencing depth past that. Like there aren't going to be any sites that have that many reads mapping to them. But you can, if you needed to, you could change that using the max depth. So you could just add in this MPileup line like -- max depth, and then tell it what you want that threshold to be. 250 is just the default for depth. So this is going to take a minute. I can actually, this is a good example. So I didn't use the and sign to start it in the background, but if I do Control Z, I stopped it. Now I do BG, sends it to the background. So now it's finishing that MPileup in the background. You can do top -U JP Damon. And oh, maybe it's already finished in the time that I was doing that. Because it looks like the only thing I have running is this and my bash terminal. So I can do jobs, and there are no jobs running. Oh, and as soon as I quit top, it said done. So that's done. And there's my BCF file output. Now there's nothing to look at there. Again, if I opened that, it would be binary, so can't really see anything. Next step is to actually take those probability calls and turn it into variant calls, and also then convert that into VCF, something we can look at. So I think all the commands are listed here, right? The only, yeah. So we're going to say ploidy1 means this is haploid because we have E. coli, it's a bacteria. -V, we're only going to have it write the variant sites. So everything that matches the genome site is going to be ignored. And then we're doing consensus calling. If you go to the SAM tools site or here, so I'll talk about let me execute this first, get it running. Okay. Now, so I'm running that last command here, number 7. What mpylum did was it took the BAM file, so the mapping, and each of these gray bars is a read that has mapped to this spot in the reference. So mpylum is going through and looking at what's the probability of, for example, this thing being an A. In our example, if, right, this is a haploid organism, and if all of our sequence came from, well, I suppose if you have lots and lots of cells in bacteria, there's a real probability that that is an A. Okay, but that's essentially what mpylum is doing, is calculating what's the probability that there's actually a variant at that site. Okay, and it's fairly low that there's a variant at that site. When we look at the VCF file in a second, right? So it's finished. You'll see that it gives us the alternatives and then some information about how likely they are. Okay. So you can look at less to see our variant call. Let's open this. Do I have it open? No. All right. So less. So this is what our variant call file looks like. So there's a bunch of metadata up at the beginning, and that it's really reflecting some of the settings, but also gives you an idea for what these abbreviations are. Okay. Like I said, so this is where it was, chromosome, in this case it's the genome reference. The position is the second column, so this gives you the start positions. The ID, which is blank in this one, because our variants don't have IDs. The reference allele, so since this is tab delimited, the rows and the columns are getting a little off here, but we're in column 4, so 1, 2, 3, 4. These, right, TAC and so on, those are the reference alleles. So the genome has that allele, and then ALT is the alternate allele. So at position 161,041, the reference E. coli genome has a T, and our sample had a G. And that has a very high quality score. So think of that as like the probability that it's correct. Okay. I don't think there's anything else that we. And in my trimmed reads, actually that's the whole file. So there are actually not very many variants in this. And you can also see that some of these, right, these are examples of indels where it looks like our sample has an extra G in there. All those ones with multiple nucleotides, those represent indel variants. Okay, so that's it. That's cool. So we have now basically this is like a resequencing project where we have defined all of the variation in our sample relative to the reference E. coli. There are cool things you can do on a diploid genome if you have population genetics. Right, there's like these programs are very flexible, and we could spend weeks talking about different kinds of data. Right, we could do diploid data, population genomic data, where you calculate the Hardy-Weinberg thing, equilibrium probability. Those things, the BCF and SAM tools have options to do that. That's why I've given you links, because so much of that depends on what kind of data you're putting into the analysis. Okay, so for those of you who are working on microbes, you could follow this pipeline to call your variants. Okay, so last thing for homework, just so that I can be sure that you are working through the genome variant analysis section, what I'd like you to do is run that whole mapping and variant calling pipeline on the original untrimmed data. Okay, so back to, come on. Uh-oh, I'm frozen. I suspect that maybe my interact session ended and now it's waiting. That's unfortunate. Okay, so my window's frozen. But anyway, you know, the original data are the SRR files. So these original read files. And I'm not saying go back and trim them. I'm saying we ran this pipeline of calling variants on the trimmed data. And I would like you to go back and see what happens if you don't do quality control on the data you get. Okay. So you'll just go through those steps we just did, except using the SRR, right? I guess maybe the only major, well, it's not even really major, but here where we had unpaired reads, you're not going to have any unpaired reads in when you use the original data. You're just going to have read 1, which is SRR 1, and this one is SRR 2. Okay, so you'll only have 2 input files when you do the mapping step with Bowtie 2. This doesn't create that. Right. It will only create that when if you named your output file that. Okay, and then I haven't decided on a due date. That'll depend on how far we get in class today with this and sort of gauging where everyone's at. I feel like people are a little behind on the BLAST homework, and I'm already overlapping the two more than I would like to. So we'll see where people are at in class today, and then I'll decide on a due date for the variant analysis homework so far. Right. The things you'll upload are the summary text files. Okay. Yeah. Still locked. And then the VCF files from both the trimmed and the untrimmed. And then in the comments for the homework, I want you to tell me the difference between the total number of variant sites between the two analyses. So what that means you're going to have to do is in the VCF file, figure out how many rows there are. I'm not sure what's going on. No, it's just locked up. I'm gonna have to quit. All right, so log back in. Okay, so it's telling me there are no. I suspect what happened was my Interact session ended while it was in less, looking at that VCF file. And that froze the, like I then couldn't exit less. So in any case, I'm logged back in now. Navigate to, whoops. Here's all of my stuff. So you're going to give me this summary and the summary you end up with after running it on the raw data. And then you'll also end up giving me the VCF files for both the trimmed and the original. So and then, right, that thing about tell me the difference in the total number of variant sites. How are you going to get that? Well, you're going to have to figure out how to count how many are in here. And that part I'm not going to tell you. Hopefully it's fairly, we have talked about how to do things like this before, but you need to tell me how many are in here and then compare that to how many you get. So each one of these, in case this is clear, I didn't say this explicitly, each row in this file is a variant site. So essentially what I'm asking is how many rows are in this versus how many rows are in the VCF file you get when you run it on the raw data, and then compare those two things. Okay. So you should also be looking at, the whole point of making you go through this exercise is seeing, what happened when we cleaned up the data. How did that affect our mapping percentage and everything? The only analysis I'm asking you to do is that, how many rows are in the trimmed versus the raw VCF. Okay. Hopefully that's clear. And I'll look forward to seeing you in class.